Yeah, good evening to all the participants. So let us start the day three, the session three for the certificate and internship course on bioinformatics. Yeah, so before proceeding to the contents of today's session, that is today's session is on about data file formats. Okay, in biological databases. And one more session is there. On tomorrow, I'll be completing all the other special biological databases. Till now, we have discussed a few databases, isn't it? So we just have a recap from your side. Like I want to check like how much you understand. On that, I can proceed whatever is uncovered. Okay. So the first question from my side is, what are the last session contents? I think we have discussed for 1 hour, 15 minutes on yesterday. We discussed in detail. Yes, very good. We discussed about biological databases. Very good. Mithul Abhinav has initiated, and even the others has can try to respond biological briefly in NCBA, and we saw some in Uniprot. Can I go for the second question? The second question is like, what are the biological databases present in NCBA. Give me some examples. See the latecomers, the late joiners. So try to uh, follow the instructions provided by Dr. Praveen Kumar Ramanujam sir. What he has mentioned, go to the go to your mail. Okay, you might have received from the Innovation Institute. You have to select yes. Then you have to go to the URL. Even myself, if you see, so I have pasted the URL. Okay, I have pasted the URL somewhere in my WhatsApp. So whenever I want, I can click and get inside. Okay, or if you click yes, obviously it will be easier for you to go to that kind of an URL. Okay. Okay. See the easy ways of use. Don't make it complex to join the meeting. Okay, I got answers once again from Mithil Abhinav that OMIM gene and about insulin protein we have discussed we have checked in various databases, isn't it? So good. So from Shreyasi gene and OMIM, you remember PubChem, gene, genome, protein, and then SwissProt. We didn't go in detail about SwissProt. I can take you to the detail on SwissProt also on today. GenBank Blast a tool is there, just we just mentioned. We go in detail in subsequent classes and gene, genome, proteins, very good. I think most of them are covered. Now, can I ask the next question? Give me some databases present in UniProt. Yeah, we also discussed about that's not just images, 2D and 3D images. These are structures, 2D and 3D structures. So the structures in the form of STF format. Okay. So what are the databases? Yes, very good. That is UniProt KB, not UniProt. That is Universal Protein Knowledge Base. The expansion is Universal. So this is the fully annotation database, isn't it? So you get more details about the protein. What else you can find the databases present in UniProt? So everyone are mentioning the same database. Can you mention some other databases present in UniProt? Like I have listed so many databases in NCB. How about in Uniprot? Uniprot confirms the sites of proteins. Okay, but still it's in Uniprot KB. Okay, so whatever sites, whatever protein-protein interactions, whatever the disease information, all these things are present in Uniprot KB database in Uniprot. Can we consider SwissProt? Actually, SwissProt and TRMBL are a part of Uniprot KB. Okay. So today I can take you. Very good. You tried well, but very good. So again, once again from Ithul, UniRef is right. UniRef meant for protein clusters. So UniProt is majorly meant for only proteins. Okay, UniProt KB, you get for full protein annotation. In UniRef, you get protein clusters of specific protein. If you search for insulin, you find for insulin. Okay, if you find for myoglobin, you can find for myoglobin. Very good. That not UniPrac, but it is UniPark. Okay, that is Universal Protein Archive. It means that Universal Protein Archive, you find only FASTA format of that specific protein. And one database which is only present in UniProt. What is that which is not present in NCBA? An exclusive database. What we discussed in UniProt, we have, you remember, we have proteome. Okay, good. So these are some of the things we have discussed on yesterday, isn't it? That now let me take you to the today's session. So on yesterday, I think I downloaded some 2D and 3D structures of a chemical compound. I believe I have downloaded curcumin. Okay, so I've taken to the NCP PubChem and downloaded 2D and 3D structure of curcumin in STF format. Can someone tell me the expansion of STF? Expansion of STF. There are so many concepts, so many terminologies, so many terms and abbreviations. If you know that, you won't forget at all. If you know the expansion of any abbreviated term, you can understand very clearly. Can someone tell me the expansion of STF? Structure, data, format. Very good. The answer is right from Shivani. Okay, now let me take you to the how to view this kind of 2D and 3D structures, whatever you downloaded on yesterday. Okay, now I'm going to open the 2D structure. Let me open in the Discovery Studio 2023. This is what I recently installed. I mean, this is the latest version I have. BioWare Discovery Studio 2023, which is a freeware. Okay, so you have to go to the, if you want to download it, okay, I suggest you to go to the BioWare Discovery Studio. So get inside, you can find free download. Don't go to some other websites. Only go to the uh, corresponding home page, and now you can register and you can download it. Okay. So after downloading, you have to install it. Okay, it occupies space up to 8 GB. Now let me take you to how to visualize it. Now we got this kind of an entry for curcumin. Okay, this is for a 2D structure. If you want to see in the form of a structure, you have to right click, show in graphics view. So whatever you see here, this is the 2D structure of a curcumin. But whatever structure you find, this can't be used for docking as well as simulation purposes. You can't use it. Only the 3D structure you have to use. Okay, so this is just for having you can take a snapshot or whatever you can make it out. But if you want to work on these kind of computation-related drug designs, try with only 3D. I'm checking where the 3D structure is downloaded. So sort by date modified. I think this is the 3D structure. So I'm going to open the same in Discovery Studio. Open it. So again, I got some kind of an entry. Right click it and show in graphics view. Okay. So this is what 3D structure. See the difference? Here you can find the conformation. So this is not a flat model. So some kind of an angle inclination is there in 3D. Whereas in 2D, no inclination. It's a flat. Okay, so you can't able to find exactly whether this ligand is binding with the binding sites or not. This is the stable conformation. In the 3D structure, whatever you see is a stable conformation. So after energy minimization protocol, okay, you find this is the stable structure in 3D environment. If you take a solid, you are generally keeping it in air, medium as the air, okay, in software environment, in a simulation environment. So whatever you find in the black window, a chemical compound is there, isn't it? So if you take a chemical in solid, it means that you keep it in air, and if it is in liquid, you are keeping it in water. Sometimes for storing or for diluting, we keep it in water, isn't it? So if you want to keep it in buffer, you have to go for simulation technique. Okay. So these are the default medium which we find in the visualizer. Okay. Now let me take you to the information of PDB, that is protein structures. So if you want to download the protein structures, okay, so the default we go for PDB, isn't it? rcsp.org/PDB. So let me take you to a protein. I download a protein and I can show you some information on that. If you see the protein data bank, okay, so you can find nearly 2.22 lakh structures are available. Whatever 2.22 lakh structures you can find is from the derived from the gold standard techniques. That is only to the experimental techniques like X-ray crystallography. I can type some gold standard techniques where for getting a three structure protein structure. Number one is X-ray crystallography, and number two is X-ray diffraction. So whatever I type here, I can also show you the same in the database. NMR, that is nuclear magnetic resonance, cryo. So earlier, if you see, if 5 years before, you find only these techniques are available for getting a 3D structure. But you now, if you see, plenty of techniques are available. Okay, most probably you can find only these four major techniques, but now you can find most of the other techniques. New emerging techniques came for getting a 3D structure for protein. Yeah, if you see this structures, okay, these are gold standard, and you can also find 10.10 lakh 68,000 number computed structure models. So how these are predicted through computation? These are predicted through artificial intelligence, through AlphaFold. Okay, so you can check what kind of a technique they have used. I'm taking you to the first computed structure analysis. Yes, the major database what we can find is the AlphaFold DB. Okay, so this is majorly utilizes artificial intelligence to predict the structure. Okay, from the already existing X-ray and NMR cold standard technique models. So we have types of scores, and based on the scores, we can able to find the precision. If the score is very less, that is less than 50, PLDDT score, quality score is less than 50, you should not use it for any computational techniques. Like you can't take it for docking, simulation, etc. So if it is greater than 90, well and good. It's equivalent to a gold standard technique predicted protein. So you can use it well for this kind of a study. Okay. So we also have filters. So first you can go for normal structures. If you don't have, you can also go for the computation. Instead of going for predicting structure of proteins, you can also take this AlphaFold predicted protein structures. Okay. So this is something related to proteins information. And now I can take you to the 2.22415 back structures. So here, if you see the structures, okay, you find so many proteins, but if you want to work on a specific protein from the beginning, I just go with insulin. In insulin, I take Homo sapiens. See, 8,585 proteins are there and, with insulin name. Okay, if I want to get a good quality protein, I have to go for refinement in resolution. Okay, so generally, the protein structures, the resolution of 2 to 4, okay, the resolution of 2 to 4, we call it as a medium resolution. And if the resolution is between above 4, okay, this is what we call it as low resolution. Okay, so instead of if you don't have any structures, you can try this. But if you have the protein with this resolution, okay, in this resolution, you can try this. Okay, that is in medium resolution. But if you have protein structures in the next range, that is less than 2 angstrom, then it is high resolution protein. This is like whatever you try with it, you get a kind of a precision results. Okay, you get a precision kind of location, site locations, if you try with the talking or simulation. So Always try with a high resolution when compared with medium or low resolution proteins. So if you want to try with the highest resolution, that is zero point 0.5 to 1.0, okay, I think these kind of predictions happened recently in the last decade. Okay, I'm going to select this as one of the refinement filter. Now we get nearly 11 proteins for insulin. So whatever you selected, two different filters, okay, you have to click refine here. If you click this arrow, so you get only 11 insulin related proteins. Okay, so one is X-ray structure of human recombinant zinc insulin. Okay, two zinc insulin at this angstrom. So let me download one kind of insulin. Okay, so now I'm going to download this structure. So don't click this download file. Okay, if you want to download the file, just get inside the PDB ID initially. The PDB ID of a protein is generally four-letter ID. Okay, just get inside, because if you click here, it download in the form of a zip file. You should not download the file as a zip file. You have to download the file generally in the form of PDB format. But there are also other formats. If you see here, we have various formats. Okay, like Gaussian software uses a GZ format. Okay, and if you see the Autodoc PYRX, it uses PDBQT format. If PDBQT format is not available, you have to convert it using conversion softwares like OpenBabel or Babelwin. This will convert PDB to PDBQT. But fortunately, the docking softwares like Autodoc or Autodoc PYRX has inbuilt OpenBabel software where it can convert the PDB to PDBQT. Then only it can go for the docking purposes. Okay. Now we want to use a common format called PDB format for downloading the 3D structure of a protein. Okay. I simply click here to download the protein. So I'm going to use the desktop for saving the files. I better can create a folder for whatever I'm going to try. Okay, I'm going to name it as like a bioinformatics demo. So I'm going to download this PDB structure as the same PDB ID name. Now, if you want to open this 3D structure, I suggest you to use Discovery Studio, or else if you have small Swiss PDB viewer, so these viewers can also be used to visualize the protein structures. Many people also try to use Python molecular viewer, that is PyMol. Okay, so Rasmol is a command line interface, but if you see this Discovery Studio, this is a user friendly. No, it's not a command line, so you can if I zoom in, okay, so like if I click and drag, you can find zoom in, zoom out. It's very easy to use if you have a mouse. Okay, so you can interact with mouse well with this Discovery Studio. So whatever you see a protein, okay, this is an insulin protein. But if you want to have a fair view, okay, you can right click here, click display style, okay, and in the protein, you have a solid ribbon, well and good. If you want to have a background like white color, I'm going to give you a white color, okay. Then what else? How can I can change solid ribbon is fine, looking good. Uh, if you don't want atoms, I simply switch off. Now see, you get a nice protein structure. Okay, this is a secondary structure protein. So whatever you see in the red, okay, it looks like in the helix structure, isn't it? So this is an alpha helix. Beta strands look somewhat different. Okay, beta strands will be in blue in color, and it looks like a standard like appearance. That's because you find a coil kind of thing. This is a helix, alpha helix. To know more about the sequence information of a protein, you can click View Hierarchy. If you click View Hierarchy, you can also find the domains of the protein, how many chains are present in this protein. You have this A chain, nearly 1 to 21 amino acid, and B chain, again with some kind of 1 to 30 amino acid, C chain. So you have plenty of chains, isn't it? So you can find all this. Other than this, you can also find heteroatoms, water. etc. Okay. In Discovery Studio Visualizer, you can find the protein information like this. But if you want to find something, like if you want to edit the protein structures, I suggest you to go to the PDB file, right click, open with Windows, right? So if you have old version of Windows, you have to open with WordPad. Don't try to open with Notepad, or don't try with uh, the Microsoft Word. Don't try with Microsoft Word. Even Notepad is okay, but WordPad is easier for you for so many uh, like for applications. Like if you just drag this WordPad, you can able to see, okay, some contents are there. Whatever you find like atom, okay, so they should be in one line, but you find this in multiple lines. Okay, if you want the atoms in a single line, I suggest you to kind of select the entire code by clicking Control A, okay, and go for a size A. Now almost you can find header atoms, everything in a single line. If not, you can still decrease the font size and you can make it up. Okay, you can go also with the font 6, something like that. Okay, so you try to find everything in a single line. If you want to zoom it, you can zoom it like this. But the font is reduced as such. This protein is still large, it seems. So the content is large. Okay, let me explain about the content present in PDB file. Number one, in the PDB file initially, still I can zoom further. Okay. So in this PDB file, initially you have header. In under header, you can find the name of the classification of the protein. The insulin protein is coming under immune system. Here, if you see the title, you get the detailed information of the protein. This is actually an X-ray structure of a human recombinant 2 zinc insulin at 0.92 angstrom. And you can see what are the chains. Okay, AC are the chains. You can find actually A and C chain insulin. You can find, and B and D insulin was purchased from Insugen. Actually, this is a recombinant insulin. They synthesize each and every chains and just got this kind of an information. And even yesterday, I think one of the participant asked me, "Sir, what is isoform? Do you remember?" So, what is isoform? Protein isoforms are formed during alternative splicing. We remember I explained you something like exons, which called as coding gene. Only the coding gene code for proteins. Non-coding genes, introns won't code for proteins. If you splice the introns and if you match all the exons, okay, not all the exons, whatever exons, desired exons, okay, that gives you a kind of a protein isoform. Okay, maybe one exon linked with the third exon can be an isoform one, and first exon linking with second exon not you find isoform two, which is not linked with exon three. Likewise, okay, you can find multiple isoforms can be formed if you have multiple exons during alternative splicing. Okay, so that is something related to the sequence. Okay, the amino acid sequences, whatever we discuss, comes under primary structure of a protein. But if you see the 3D structure, okay, the 3D structure of a protein, if you edit in WordPad, you look the information like this. If you are going for a kind of a recombinant protein, okay, so definitely under organism underscore scientific, you can find what is the exact species. The species name is Homo sapiens, no doubt in that. And you also have expression underscore system. Okay, this is what the species they have used for genetically engineering. Like if they want to produce maximum insulin, they try it with Homo turtella pastoris. So this is the species they use. So defaultly they use E. coli, but sometimes they use the other expression systems. In this case, okay, in this 3D structure of a protein, they have tried with this kind of pastoris kind of bacteria. And if you see further, okay, so who are the authors initially got this 3D structure of a protein? And when they got, in 2017, I think they have released this. Also, they published in a journal called the name of the journal is mentioned, resolution is mentioned. Okay, so some information of that you can find, but more important information other than header and title is the atoms, which is present after this information. After this scale, you can find atoms. So whatever you find atoms for nothing but the amino acid atoms. A protein is nothing but a building block of amino acids. Okay, so if you see this coordinates, okay, under atom you can find 1, 2, 3, 4, 5. So for the glycine, if you see the glycine, okay, which is the first amino acid, how I am telling glycine is the first amino acid, glycine 1. So up to here is 1, isn't it? So under in A chain, you find glycine as the first amino acid position, and whatever coordinates you find here is the X, Y, and Z coordinates of that specific atom called nitrogen. In glycine, if you have an atom called nitrogen, the specific position for a stable protein. Okay. As I earlier mentioned in Discovery Studio, I opened a chemical compound of 2D structure and 3D structure. The 2D structure is flat, but the 3D structure, you find a kind of a conformation. You find a kind of cis and trans, some kind of stability. The conformation gives you the more stability. 1 minute, I can check any participants has to join. See, it makes you to halt you for some time. Okay. So the participants, I suggest you uh, the participants, the latecomers, I suggest you to click yes in the email ID received from Innovation Institute. Then, if you try to click the URL, it directly take you, it admit you. There is no need to explicitly admit from my side. Okay, let me take you to the explanation of the PDB. So this is a PDB file. Okay, the insulin structure PDB file. I was explaining something about the coordinates. Okay, the important coordinates present in insulin protein. So the first amino acid. Okay, the first position amino acid you can find glycine. Okay, and this is the A chain. And the second position amino acid is the isoleucine. You know the structure of amino acid. In amino acid, you have a chiral carbon atom at the center, okay, and you have alkyl group at the top, and amino group in the left hand side, and carboxyl group in the right hand side. Okay, so this is how you find, isn't it? But you have plenty of atoms. So these are the functional groups, okay, molecules. However, the atoms. So here you can find this is the C alpha position. This is the C alpha position. Actually, uh, the first atom is nitrogen. The second atom is the C alpha. Third atom is just the chiral carbon atom, which is present in center. O is the oxygen atom. H1, H at first position, second position, third position, and of course H alpha 2, H alpha 3. So there's if it is linked with carboxyl group, okay, whatever H you find, okay, this can be given like this also. Even in alkyl group, you can find this alpha 2, alpha 3. Okay, now we got some information about the coordinates. Okay, so these coordinates, like if you want to go for a docking, and if A and B chains are most important, and if you want to ignore the other chains, okay, so you just have to, you can also edit the protein by selecting here from C. I'm selecting the C now, D, selecting C followed by D. So just dragging, okay. So up to this, I'm going to select. So this is the termination up to TER, you have to select it. And if you don't want the C and D, just delete it. Just delete it. And from here, you can able to find the other atoms also. After atom, you can find HET-ATM. What is HET-ATM? These are heteroatoms. In the heteroatoms, okay, you can find the metals. Okay, if it is a hemoglobin, you can find iron as the heteroatom, and you can also find water molecules as the heteroatom. Okay, so if you don't want heteroatoms, especially water molecules, because if water molecules hinder the binding sites, if you want to remove it, even you can select it. Okay, you can select the water molecules up to here, up to the HOH. This alone you can select it and it can delete it. Because if you delete something like zinc and etc., it disrupts the 3D structure of a protein. These are already attached to a protein. You can't able to delete a metal atoms as such. But the water molecules you can remove. Okay. It depends on what kind of a docking you're making. And if you want to save this file, okay, save as this file. So I'm going to click File, Save As, select the plain text document. That's the plain text document. So I'm going to save this as underscore modified dot pdb. I give some file name dot pdb in double quotes. Okay, so whatever I converted, like deleting C and D chains and also deleting the water molecules, I have saved it as file name dot pdb under text documents in double quotes. This is how we can save it. Now, if you get a prompt like yes or no, you click yes. That's it. Okay. So I think some participants raised some comments. In which format was the file downloaded? Can anyone please tell? It is in PDB format, .pdb. So what I've taken is I've gone to the particular insulin protein, click download files, and I click PDB format. So in the PDB format, when I opened in Discovery Studio, so I got the 3D structure like this. And when I opened this in the WordPad, I got like this. These are the coordinates. Now, how to delete some unwanted coordinates? That's what I showed you. You can either make it up using the WordPad, or else you can make it out using the Discovery Studio. So if you want to delete something like a C chain, okay, I select the C chain, entirely cut. Okay, but it's wise to do using WordPad, because some of the coordinates will disrupt you. It won't permit you to go for MD simulation. Okay, so you have to try well, because if you save this file, obviously it's saved like a Discovery Studio coordinate PDB file. You won't get something like a header title, like however you downloaded as an X-ray structure. Okay, so if you want to go for a simulation, even you can, it's better to use a WordPad for editing the coordinates. Or if you want to go for simple kind of a docking, something like that, you can try even here. Whatever you don't want, okay, the D coordinate, if you don't want the D chain coordinates, you select the D and you can try to delete it. I'm selecting the D. Yes, I've deleted, isn't it? So you have multiple D. This is like actually a uh, heteroatom, a compound binding with a protein. Okay. So if you don't want water molecules, select it, but it's already deleted along with the chains, it seems. So you don't find. So some kind of confusion will be there. Okay. If you try with the Discovery Studio. So I suggest you to go try to use the WordPad. Okay. Now let me explain about other formats, especially one participant has raised, like, sir, can I mention something like Swissprot as one of the database present in UniProt? You know, the UniProt is a fully annotated database. Defaultly, it takes you to the UniProt KB database. UniProt is a very large URL. Okay, you find plenty of databases. One such database is UniProt KB, a fully annotated database. If you want to type a particular protein, you are going to type a protein here. Okay, you are getting some information of that. Yesterday also, I think a participant has raised me like whether we can download all the sequences. Okay, like for example, if I want to select 10 proteins, whether these 10 proteins can be downloaded in the form of a single file. Yes, for this we can try like, if I want to download these three files, that is insulin receptor, insulin degradation file, degradation enzyme file, and also insulin ligand file in a single document, click select all these three, download selected, okay, download as FASTA canonical. So it's in compressed format. Do you want in compressed format or no compressed format? If you want to use as such immediately, go for no compressed. Click download. Now, whatever you get, okay. So this is a FASTA file of insulin, three proteins, isn't it? So three different proteins. So I'm going to download this as such. Now you can check it out. So whatever you downloaded, the three proteins. Okay, if you open it in Notepad, you remember yesterday I told you, na, better use a Notepad for visualizing faster. So after one protein, immediately after you get a kind of a greater symbol, and you can find the second protein information. So one, I think this is first, this is second protein, and this is the starting of the third protein. Okay, this is how you can download multiple sequences. Select this, download this. And if you're going for pairwise alignment, you select even multiple and click this blast. So more about the blast tool we see in the blast session. Okay. So I think I've cleared the doubt what Dio Krishna, a participant, asked on yesterday. Now let me
Yes, sir.
explain. Good. Now let me explain about the category of databases present in Uniprot KB. That is, we have reviewed information, which we call that as SwissProt. If it is unreviewed, we have TRE. When I search for insulin, I got this much results. 3 lakh results I got. But out of 3 lakh results, or the reviewed information you find very less, that is up to 5,342, 41, which comes under SwissProt. Okay, so what is SwissProt? Even if you search in Google, okay, SwissProt is a separate kind of a. You can also find a separate URL for SwissProt. Yeah, this is actually a SwissProt knowledge bases.
Hello.
Yes, sir. Yes.
Sorry for disturbing, sir. Sir, I want to download like if I want to download only the rodent insulin PDF
Mhm.
file or amphibia or like mammalia. So how I can download? How can I
Mhm.
do that, sir? Can you please show me once?
Yeah, you have to select the rat alone. Okay, even I didn't select any advanced filter like human. Default it lists you the human. If you want the rat, okay, so just go to the ratus norvegicus. It's even simpler for us. Okay, select that whatever ratus norvegicus you have, or you simply go to the rat. If you click the rat, you get only the rat kind of instant proteins. How much you can get? Nearly 1982. But whatever you want to try, that alone you can select it and you click down. Okay,
No,
so
sir.
this is how we can.
Sir, I want to download phylum-wise protein structure, sir. So is it possible to download
What
this
what
and after
what protein?
phylum-wise, like amphibia, arthropoda, mollusca, this kind of thing. So from arthropoda, mollusca, bacteria,
No,
this
no,
kind
no,
of.
no, no, no. See here, no, no, no. See, your voice is not feeble. Okay, some internet strength, internet strength is there, freaking often. So I suggest you to type the question in the chat box. Type the question in the chat box. I can answer you. Okay. So now, meanwhile, I can proceed explaining about the left out concepts. Okay. So if internet is not good, okay, I suggest you to type the question in the chat. Now let me explain about that SwissProt and TREML. For a SwissProt, you have a separate kind of URL also. Okay, so actually the SwissProt website, okay, this is majorly present in Switzerland. And Uniprot, this is Uniprot is not just from a single country, England, that is maintained by London, UK. It's also maintained by Germany. Okay, so the Germany is collaborating with the DREML database, and the next country is the Switzerland, which is having the SwissProt database. So three different countries joined together, okay, to form a Uniprot URL where you can find plenty of databases like Uniprot KB, Proteomes, Uniref, Park, etc. Got it? So this is a combination of three different informations. Okay, three different countries came together to host this kind of a large database, a fully annotated database. Okay, So now I also explained you like what kind of how to download the multiple sequences and what kind of format you can download for various applications. So the whatever format I downloaded, okay, in the FASTA format, this can be used. This can be used for multiple sequence alignment, phylogenetic reconstruction, etc. Okay, so that is multiple sequences in one FASTA file can be used for multiple sequence alignment and phylogenetic reconstruction. Okay, now let me take you to the next concept. So if you see the thing, okay, what we discussed about just the sequences alone and structure. I showed you one thing that is PDB, like how to edit everything. And now I can take you to the some other databases like KEGG, the most important database, KEGG, that is Kyoto Encyclopedia of Genes and Genomes. We have lakhs of biological databases to use, but one has to select, okay, based on the kind of a research what you want to do, okay. So based on the type of a research, you can use even exclusive databases. Now, whatever database I show, NCBA, UniProt, these are the default things you must use for all the types of research. KEG is Kyoto Encyclopedia of Genes and Genomes. This is majorly taken care by Japan. So only you'll be getting an URL like genome.jp. Okay. So in Cake, you find plenty of databases. So the most popular and the largest database for pathway you can find in Cake, Cake Pathway. Okay, like Cake Pathway, other popular databases are Cake Genome, Cake Compound, Cake Reactions, Cake Enzymes. So you can find more details, specific details in this cake. So let me explain one by one. First, I'll take you to the cake pathway database. In cake pathway database, you have classification of databases. If you're working on pharmacognosy field, that is using a natural plant compounds to treatment of diseases, okay, you can also find in the human diseases, or if you want to find the secondary metabolites from the plants, you can try this kind of a pathway database. Like how the secondary metabolites are formed in the pathways. So that information you can find here. Okay, like not only the plant compounds, even if you want to check with the carbohydrates, if you want to check with each and every amino acids, you have a detailed pathways, how the amino acids are formed, how the biosynthesis happens. Okay, and if you want to check with human diseases, you want to go with the genetic information processing, cellular process. Let me explain at least one. Go to the human diseases. If I go to the human diseases, you have plenty of cancer, you find infectious diseases, bacterial, viral, parasitic, immune diseases like autoimmune diseases, neurodegenerative. So the most important diseases where the human population is majorly suffering from, okay, that kind of diseases are mentioned here. Okay. So, for example, if you're focusing on a specific cancer, okay, it can be a thyroid cancer, okay, or the most popular cancer in the India is breast cancer. Okay, many people are facing this. So let me take you to this kind of a breast cancer. So if I take you to this breast cancer, okay, it takes you to a detailed pathway. Okay, a detailed pathway of the breast cancer. So at the end, you can find what kind of a symptoms, and for the symptoms, what proteins are responsible. So here you can find so many boxes and you find some kind of an abbreviations, isn't it? If you want to know more about this, click on one particular box. I'm going to click this ER box. If I click the ER box, okay, you get some information here also. Here also you can find some information now. Yes, this is the estrogen receptor 1, which comes under estrogen receptor alpha. Okay, so this is a major target receptor for breast cancer. So what are the different pathways linked with this protein? Okay, multiple pathways are there. Okay, it can take you to this bright okay website. Okay, so bright database. Okay, and you can find the amino acid sequence of the specific estrogen receptor. You can also find disease informations. disease targets, what are the best drugs used for that particular target. Okay, so these are the popular drugs used in the market. So whatever popularly used in the market alone will be shown. If something is banned or if something is prohibited, this will be removed automatically in this website. Okay, so you can find more major information of the drug interactions, protein-protein interactions, different pathway link, all this information in this cake pathway. Okay, so I just click the only one box. There are so many boxes. If you want to link with the RAS pathway, okay, if you want to check about the map cake, you can't able to try with all the pathways if you're working on a particular cancer. You have to try with only one pathway. Okay, that itself it looks like a PhD research work. Okay, a PhD project will be a one pathway and one target protein. That itself will get more kind of information, like a protein interacting with other proteins, and you can work on that by try to find the gene expressions. So, okay, so you can find nearly some minimum 12 to 15 pathways also for this cancer alone. Then you have to see in which where the research is not extensively studied. You have to select accordingly. You have to check first initially through the literatures. If one is not explored in detail, you have to select that pathway and you have to go to the protein and you can work on that. Okay. So you have to check for the novel proteins and novel pathways, whatever well established, like MAPK, phosphoinositol 3 kinase, all these things are multiple researchers around the world have tried in various ways with various drug delivery techniques, etc. Okay, so you have to check for the novel protein targets and novel protein pathways. Okay, so this is something related to the pathways. I have taken to the disease pathway. So I can also provide an example for metabolism pathway. You know, glycolysis. I think in Inter, that is on plus one and plus two, many have studied in the school level itself about the glycolysis. So remember that if you see that, okay, the initial the initial input, what we take is the glucose, isn't it? And the final output is the pyruvate, isn't it? So you find only 10 major steps. But here in this cake pathway, see, so many sub-steps are there. So if you want to convert the glucose to glucose 1 phosphate, whatever mentioned here is the enzyme commission number. Okay, if you click this, it gives you the detail of the enzyme. Okay, so you get a detail of an enzyme, but not only you get the major 10 steps, you also get some sub steps. These sub steps are not mentioned. Did you hear something about arbutin, arbutin 6 phosphate, salicin 6 phosphate in the glycolysis cycle? So these are something linked, interlinked with the glycolysis itself. It's not coming under that pathways also. Okay. So, you know, the pathways are also interlinked with each other. Like glycolysis can be linked with gluconeogenesis, can also be linked with citric acid cycle. Okay. Even if you want to see the linking of these pathways, you can able to see in this cake path. Okay. So, now just now we can able to see this kind of detailed information, like however I showed you for the breast cancer pathways. Okay, so for estrogen receptor. So you can scroll down and you can find some information on. Okay, so likewise, okay, you find extensive information in this cake pathways. So other than pathways, you can find also disease. If you want to find some more information of the disease, if you want to find the drug information, if you want to find the compound that is again other than the popular drugs available, synthetic or the what we call it as allopathic drugs. Okay, if you want to find something related to plant compound and whether the plant compound can be used to target a specific target for a disease, all this information also you can find from the compound. Check genes is also a popular database. Okay, so you can try one by one. For example, if I try with a compound, okay, so you have so many compounds listed here. You can type the compound name. For example, I can type aspirin. Okay, click go. You can give whatever you want. Curcumin. Yeah, we have tried with curcumin, isn't it? You can also try that. Okay, so you have to click entry to get more details of that. So, like, however you found for the pathways for the particular receptor, even for the compounds, you will find so many information. Even you can download the information. One day that if you want to download the coordinates in mol file, you can use this database for downloading. So you can download a small file, but in NCBI PubChem, you can download in other formats, SDF and other formats. You don't find mol file. Okay, like in some kind of docking softwares, it asks you a mol file or mol2 file. So in that case, you can think about to use these kind of a databases. Okay, like aspirin, you can try with a curcumin, some plant compound. Yeah, in curcumin, you have even the derivatives of curcumin. Okay, nearly six compounds are here. Okay, if you want to download a specific mol format, you can select and you can download. Okay, so likewise, you have reactions. So many reactions are interlinked, isn't it, for the biosynthesis? Okay, so we have to type a reaction. Okay, so for this, examples are mentioned. Okay, go to the examples. So some examples are mentioned. Try to check it, and you check for your research. Because I am poor in chemistry. If you are if you are a chemist, so you can check the kind of reactions, and you can try this. Okay, so or like if two reactants are reacted, what kind of products will be formed? So related to the biological reactions. This all this. This is related to the biological reactions. Okay, what we call it as biotransformations. In pharmacokinetics, we have biotransformations, isn't it? So more about the biotransformation reactions you can find in the reactions. So like KEG, okay, we also have other popular databases. There is a database called Gene Cards. This is the largest database for genes. Like, what is the major difference between the NCBI gene and gene cards? In NCBI gene, you get the gene information, like a sequence information. But if you don't know anything about a gene, if you want to go for a gene annotation, like how I go for protein annotation using UniProt KB, okay, the gene cards is similar to that. This is for gene annotation, like UniProt KB gene cards. If you are typing for a specific gene, okay, for example, just now we have gone for the breast cancer protein, isn't it? So BRCA1 is one of the uh, important gene responsible for causing the breast cancer. So I just typing the PRCA1 gene. Now we can get some information on that. Okay, what is PRCA1? And so more information of the gene. So if you drag C here, from here to here, you have so much information is there. Like Uniprot, you get detailed information like what are the gene-gene interactions, gene transduction informations, the chromosome location. Okay, chromosome 17, you can find the gene. Okay, and about the locus and about the types of disease. It doesn't mean the BRCA1 is just causing the breast cancer. This is responsible for causing other types of feminine cancers. Okay, there are ovarian cancer, cervical cancer. How about the expression in that types of cancer? What kind of literatures are recently, the researchers are done? What are the recent studies happened? And how about the protein structures? Okay, well, what is the PDB-ID? Okay, so all these informations you can find in detail in gene cards. Like Uniprot, you can find gene cards as one of the important database for genes. Like this, okay, we have databases like TR, okay, and whatever is uncovered, okay, so that of course from Uniprot itself it link you to the TR, but TR is of course a separate database, okay. Even you can go to the EMBL. So see here, EMBL is there. EBA.ac.uk, you find yeah, it is linked with the Uniprot and it of course linked with the TR-EMBL. For the EMBL, we go with the European Bioinformatics Institute.ac.uk. There you can find this EMBL database. EMBL is generally for gene sequences. TREMBL for protein sequences in EVA. Got it? So this is for gene, that is for protein. Now I let me take you to the PPT. So up to what we have completed in databases and tools in NCB, I think this we discussed in the last class itself. But more about the tools, blast tools we see in the blast session. But we have seen something about databases like nucleotide protein, etc. So literatures we can go with PubMed or Medline. PubMed for biotech literatures, Medline for medical based literatures, that is clinicians rated literatures. Then NCBI Chenbank, how the window looks like, what is the URL, and NCBI PubChem, how the window homepage looks like. So you can either type the compound name, drug name. Not only that, you can also use a keyword like a receptor target. EGFR is epidermal growth factor receptor. Even you can give a protein target. Okay, if you give a protein target and if you click enter, what you will be getting, what are the drugs used for targeting a specific target? You get all the list of drugs. Okay, even this is important. Like for example, when we worked during the pandemic time, okay, when we try to get a best drug for SARS-CoV-2, that is for COVID-19 infection, the most important target is the spike glycoprotein, isn't it? So if you type a spike glycoprotein, okay, so obviously you get a list of drugs. So you can download all the drugs in the form of a single data set, and you can also try to go for virtual screening. That is, I can tell you once again, I take you initially to the NCEBA PubChem. I better close the WhatsApp. Take you to the PubChem. So in the PubChem, I give you an example like EGFR. You also have COVID-19. See, these are actually already inbuilt. If I click COVID-19, you find this much drugs are there for treatment of COVID-19. 1,733. These are the drugs used in research actually for treatment of COVID-19. So if you want to download all the drugs, simply click download. Instead of 2D coordinate, click 3D coordinate. Okay. So because it's a large file, 3D coordinate of this much compounds, I suggest you to have a compression format. Okay. So even okay, this is not bad. So, but you have to remember, you have to set the 3D. This is most important. And try to click SDF. So all the 1,700 structures will be saved in a single SDF format file. Okay. That you can use it for virtual screening using Autodoc Pyrex. If you try with it, you can come to know which is the best compound for uh, a specific target. So COVID-19 is a disease. SARS-CoV-2 is a virus. Spike glycoprotein is a specific target. Okay, but there are also targets like polymerases, proteases. If you want to try for the what is the best drug for proteases, polymerases, you can also specify the specific protein name. Okay, like for example, EGFR, epidermal growth factor receptor. You have so many inhibitors. How many inhibitors? 261 results are shown. You can download that and you can check which is the best one. There, this is the important target for most of the cancers. For almost all the cancers you can find, this is one of the important target receptor. So even using the target receptor, you can download the chemical compounds. Okay. So not only that, we also it gives the pathway information, cell lines, biosis, more detailed information you can get from that. Okay. So you can either give a disease name, you can give a target name, you can give a compound name, you can give a drug name to which can consider as a keyword to search of drugs. Uniprot KB, I think we have completed how to search a protein in Uniprot KB and how to search a 3D structure using PDB is completed. NCP MMTB. So in NCP, you have to select structure, and if you type a protein in, it takes you to this MMTB website. You can able to visualize the 3D structure of a protein, okay, in the inbuilt visualizer. You can download the sequence, you can download the 3D structure, okay, and you can interact with all this information. AlphaFold, of course, this is you can find plenty of databases or linked with the AlphaFold database. Okay, even in RCSP PDB, you can find one AlphaFold. Nearly more than 10 lakh structures are available. I told you already, if you have PLDDT score, which is predicted local distance difference test score, if it is above 90, the quality is very good. But if it is less than 50, don't take that. The quality is very bad, so you can't take it for any completion study. Okay, so this is something about AlphaFold. Okay, biological pathway databases. I have taken you to the cake pathway. Okay, so if you want to go for a hepatocellular carcinoma, for the hepatocellular carcinoma, a detailed disease pathway information is shown. Okay, the left out is something related to the special databases like protein-protein interaction databases, gene-gene interaction, drug-protein interaction databases. So this alone is left out. Okay, so to know more about that, okay, we'll be discussing in the next session. Okay, and I think we also covered the different file formats. You remember the file formats? File formats like generally if you go for GenBank, you find locus name, accession numbers, GenInf ID, PubMed ID. Even for a sequence, you have a PubMed ID, not only for the journal purpose. So locus names, how the locus names looks like. If you want to get a human beta globin, you can type like HumBB. Okay, even that is a keyword, you can use it. Or you can, if you know the accession number, type the GenBank accession number like this with a version number. Or you can also type the gene info ID like this. Okay, or also you can type the PubMed ID. So you can give any of these identifiers to get a particular protein information. And bioinformatics database formats, FASTA, this is the commonly used. GenBank is available in NCBI. SwissProt is available in UniProt. In UniProt, you have a SwissProt format like this. So you have ASN.1, XML. These are other formats present in various databases. So FASTA format, how it looks like, creator and the identifier and the sequence information. This is a no annotation format. And GenBank format looks like this. And SwissProt format looks like this. Is how a SwissProt information for a protein structure looks like this. Okay. This is not for a structure, this is for a sequence. For a sequence, you get like this. And XML format looks like this. This is a kind of a database. Start with XML tag, and you find so many user-defined and predefined tags inbuilt. Okay, and if XML is linked with HTML, we call it as an XHTML because XML is a database, HTML is a design language. If you want to web page design language, you use HTML. If you want a database, you can use XML. You can link XML with HTML in the form of .xhtml. Also, you can include a style sheets and linking sheets. Okay. So I think I've completed even data file formats. Other special exclusive databases, biological database, we see on tomorrow. In which format was the file downloaded? That is explained already. Sir, what is the difference between PDB format and PDB gz? Actually, PDB gz refers to the Gaussian format of the PDB. Gaussian, you have a software called Gaussian. Okay, so if you want to work with Gaussian software, then you have to use Gaussian PDB. Can't use normal .pdb file. Okay, any other doubts? Where can we check the toxicity profile? Actually, these are generally the toxicity profiles. You can go for toxicity, like toxic, I think, I believe. You can go for pre-admit. So this also predicts you the toxicity of the compound. Go to the pre-admit tool, you find the toxicity. Okay. Give the 3D structure or just feed the canonical smiles format and get the toxicity of the compound. And any other thing? Pre-admit is the tool. Okay. Then we see the left out concepts that is on the special biological databases on tomorrow. So thank you for your patience and interaction. Have a great day.